Okay, so now we are moving on to talk about gluconeogenesis. So here we'll talk about the pathway of gluconeogenesis, of course, and then we're going to spend some time about the different precursors, where they come from, how they feed into this process, and keep in mind that you have to kind of think about we're mostly going to be in, of course, the fasting stage for gluconeogenesis. And so these precursors are going to be byproducts typically of other cells doing beta oxidation or breaking down of proteins for energy. We'll talk about lysine and leucine with respect to gluconeogenesis. They are not going to be able to be precursors for gluconeogenesis. You can't make glucose from them. We'll also talk again about the Cori cycle as well as the alanine glucose cycle, which is going to be important for the precursors of gluconeogenesis. Okay, so in general, during fasting, your liver is going to be given the job of maintaining blood glucose levels. And so your liver is going to have to reverse glycolysis. And this is a process called gluconeogenesis. The main precursors are going to be lactate, glycerol, and amino acids, particularly alanine, which we'll see why. There are going to be three main steps that are going to be reversed. And these are your pyruvate to phosphoenolpyruvate. So pyruvate has to go back and enter into glycolysis, of course. And then your fructose 1,6-bisphosphate has to reverse to fructose 6-phosphate. And then, of course, your glucose 6-phosphate has to become glucose. So during your fasting stage, there's going to be your blood glucose is going to be spared for your red blood cells, your testes, as well as the kidney medullary cells. The brain also will use high amounts of glucose, depending on the amount of time of fasting, of course. On a long-term basis, most tissues are going to require glucose for functions such as the synthesis of ribose through that pentose phosphate pathway. All right, so here is glycolysis versus gluconeogenesis. And so you can see, we'll start at the bottom here, blow this up a little bit. So you see that we have pyruvate. So in our glycolysis, we have pyruvate, and it goes to acetyl-CoA through pyruvate dehydrogenase, and then you enter the TCA cycle and you get lots of energy. Now for gluconeogenesis, you still also have pyruvate. However, during the fasting stage, remember, in the liver, the fatty acids or the amino acids are going to be what feed into the TCA cycle and generate energy. Your pyruvate is going to be a precursor for gluconeogenesis. And pyruvate is going to not come from glycolysis in this case, but instead is going to come from precursors of gluconeogenesis, such as lactate, alanine, or other amino acids. And then in the mitochondria, as we talked about before, you have pyruvate carboxylase. Pyruvate carboxylase, remember, takes that 3-carbon pyruvate and makes it into a 4-carbon oxaloacetate. And then oxaloacetate itself can become phosphoenolpyruvate through an enzyme called PEP carboxykinase, and that's shown here. And then once you get phosphoenolpyruvate, you can go through glycolysis and reverse many of the reactions of glycolysis until you get to fructose 1,6-phosphate. And then fructose 1,6-phosphate, you have to reverse the glycolysis phosphofructokinase 1, and you do that using a fructose 1,6-biphosphatate, or bisphosphatase. And then that will lead to glucose 6-phosphate eventually, and then you have a glucose 6-phosphatase, and that will lead to the release of glucose. Okay. Oh, this got kind of wacky, sorry. So basically, after a few hours of fasting, your glycogen begins to be degraded by glycogenolysis. right? So during the fed state, of course, you are going to pick up glucose through dietary sources. Through the fasting stage, you are going to get your glycogen broken down. Glycogen broken down, and that's going to provide you with glucose. Now, as your glycogen stores are going to decrease, of course, if you eat again, you're going to reset this whole cycle again. But during like an overnight fast or a prolonged fast, your glycogen stores will decrease. Your adipose tissue tags are also going to be degraded and released, and these are going to be a fuel for muscle and liver cells, of course. But they're also releasing glycerol, and glycerol is a precursor for glucose, as shown here. And then you're also going to have amino acids that are going to be released from the muscle. Your muscle is going to use branched chain amino acids, especially as a source of fuel during fasting stages. And then they're going to release alanine, for example, and alanine is going to be a precursor for gluconeogenesis. In addition, of course, you always have lactate being produced during both the fed and fasting states by your red blood cells. So those are always going to provide carbons for gluconeogenesis, especially in the fasting stage. Now, after about 30 hours of fasting and you're starved, so we'll put this as 30 plus hours, your glycogen stores have mostly been depleted, and gluconeogenesis is the only source of glucose. So you have gluconeogenesis. So remember that in the fasting stage, if you add alcohol to this scenario, keep in mind that alcohol will provide energy, but alcohol is going to shut down gluconeogenesis. So alcohols can quickly become hypoglycemic in this situation. Just wanted to point that out. Okay, so let's just put this all together. We have mobilization of your nitrogen and carbons during fasting. So we're going to talk about kind of the big organs. So we have on one side, we have our muscle cells. So we have a muscle. Okay, and our muscle cell is going to break down especially your branched chain amino acids. So it's going to take your amino acids and it's going to use the carbons and it's going to go into energy. But it has to do something with the nitrogens. And so in the muscle, we'll talk about in a future lecture how this happens, but in the muscle, when they start using amino acids for energy, the nitrogen has to be released and picked up. And so our nitrogens are going to be transferred to alanine, or make alanine, or our nitrogens can form glutamine. Now, alanine will be released into the blood, and glutamine will be released into the blood. And what's really important about these two is these two are really the ones that are going to move around nitrogen during the fasting stage, both alanine and glutamine. They kind of had different fates. Alanine will donate directly to the liver. So we'll have our liver. So the liver will pick up alanine. And alanine is a precursor for glucose, for gluconeogenesis. Now we also have, so I'll put this one too. We're going to build on this. Okay, so we also have glutamine to deal with. So glutamine can also go to the liver. Okay, and so glutamine can also feed into gluconeogenesis. The other place that glutamine can go to your small intestine. And in your small intestine, glutamine will become alanine. And cells in your small intestine can do gluconeogenesis. Or you can get energy, and then you get excretion of your nitrogen in urea, for example. Now, the other thing that glutamine can do is go to your kidneys. So here's your kidney, and kidneys will pick up glutamine. And then glutamine for your cells in your cortex can do gluconeogenesis. Or your nitrogen can buffer urea. Now, the reason I did a dotted line for glutamine to your liver is because that's not the major route of glutamine. The major route of glutamine is to go to the kidneys or to go to the small intestine, but it can also go to the liver. Now, alanine created by your small intestine can also go into the blood, and this can go back to the liver, and you can have alanine. Your red blood cell, of course, is going to pick up your glucose. So you have your red blood cell, and it's going to pick up glucose, and then it's going to generate lactose, lactate, and this is going to go into the blood. And lactate is another precursor for gluconeogenesis. Now you also have your adipose tissue. Yeah, your adipose tissue is releasing fatty acids. And fatty acid is going to create energy. It's also releasing glycerol. And glycerol also can be a precursor for glucose. It comes in at a little bit of a different point than your alanine and your lactate. And so it enters the process a little bit further down. But so basically this is what's going on in your fasting stage. And you have mobilization of both nitrogen and different carbon sources. The nitrogen is going to have to be dealt with, and it goes through, of course, urea cycle. And then eventually you're going to get excretion. And the carbons are going to be used, many sources of carbons are going to be used to make and maintain glucose levels in the blood. So gluconeogenesis is energy costing, but the energy, of course, is going to come from beta oxidation of fatty acids. You have 2 moles of pyruvate that are going to generate 2 moles of 1,3-bisphosphoglycerate, and that's going to require 6 ATP to do that. So again, the major substrates or the precursors for gluconeogenesis are going to be glycerol, lactate, alanine, and pyruvate. In the mitochondria, the pyruvate is going to become oxaloacetate. Oxaloacetate, remember, cannot leave the mitochondria, so you have to do a transamination reaction to malate. And then in the cytosol, malate has to become oxaloacetate again. So lactate is going to be produced by exercising muscles or always by the red blood cells. Glycerol is going to be released from adipose stores, as we just talked about, and the carbons from amino acids are going to come from the muscle cell. Alanine is going to pick up nitrogen from other amino acids the muscle cell is using for energy, and it's going to bring that nitrogen to the liver so the liver can process that nitrogen through the urea cycle and then use the carbons from alanine to make glucose. So the carbon sources for gluconeogenesis from pyruvate, and again, the amino acids feed in through pyruvate, different intermediates of the TCA cycle, and there's interchanging between both glycolysis and gluconeogenesis. So again, here's our picture. So now we have our different amino acids, and they're going to become pyruvate. We have the alanine, which was released by muscle cells. We have lactate, which was released by different cells doing anaerobic glycolysis, and they're all going to converge onto pyruvate. And so pyruvate through pyruvate carboxylase will become oxaloacetate. And then you have a phosphophenol pyruvate carboxylase, which will generate PEP, phosphoenolpyruvate. and then this will go on to form eventually glucose. And you can see glycerol over here will enter gluconeogenesis by generating glycerol 3-phosphate, which then can feed back in through glycolysis to eventually form glucose, but it bypasses the whole mitochondria portion of gluconeogenesis. Okay, so in the liver we have, of course, your lactate dehydrogenase, and so your liver will pick up lactate from the blood, and through lactate dehydrogenase, which is going to reduce NAD, you generate pyruvate. Now for alanine, of course, is going to be converted to pyruvate as well through an aminotransferase, and so this requires your alpha-ketoglutarate to glutamate, and then you're going to generate, again, pyruvate. And just keep in mind that your nitrogen, in this case, will enter the urea cycle. Okay, so we also have glycerol, another precursor, and our liver cells has glycerol kinase, and glycerol kinase will form glycerol 3-phosphate, adds that phosphate group to it, and then you get reduction of NAD to generate dihydroxyacetone phosphate, and that's through that glycerol 3-phosphate dehydrogenase. Same thing that happened in your glycolysis, but just reversing it. All right, so we have here our dihydroxyacetone phosphate. We also have our oxaloacetate, and we generated pyruvate. So these are all TCA cycle intermediates are going to be the carbon sources for our glucose. Now just keep in mind that we have propanoate, which, remember, was a derivative of fatty acid oxidation. So remember you had beta oxidation, which generates acetyl-CoA, and then if you have an odd chain fatty acid, you have propenyl-CoA that gets developed, and it's a 3-carbon. And then this gets converted to methmalonyl-CoA, and this is rearranged to form succinyl-CoA. And so acetyl-CoA from beta oxidation of fatty acid cannot form pyruvate. Acetyl-CoA is going to be used in the TCA cycle to generate energy. The succinyl-CoA that's generated from propenyl-CoA can be used in gluconeogenesis because it can form malate. And remember, malate can form oxaloacetate. So in beta oxidation of fatty acids, your odd chain fatty acids can provide carbons for gluconeogenesis, whereas your even chain fatty acids will not provide carbons for gluconeogenesis. All right, so let's start talking about gluconeogenesis. We have what are called 3 bypass reactions. We're going to take pyruvate to phosphoenolpyruvate, or PEP. We're going to reverse fructose 1,6 bisphosphate and generate fructose 6-phosphate, so we have to remove one of those phosphates. And then we're going to take glucose 6-phosphate to glucose. So again, we have to remove a phosphate. And you have to remove that last phosphate, or else glucose 6-phosphate would never cross the membrane, but glucose will. So this is bypass 1, our pyruvate to phosphoenolpyruvate. This is going to take 2 enzymes to do this. And so in glycolysis, keep in mind that pyruvate is formed from phosphoenolpyruvate via pyruvate kinase. And in gluconeogenesis, they're going to have several steps to reverse this reaction. So in this case, you have, again, pyruvate going to oxaloacetate through pyruvate carboxylase, and then oxaloacetate through phosphoenolpyruvate carboxykinase to phosphoenolpyruvate. And keep in mind right here, it's very important that this does not use ATP, but rather it uses GTP. Okay, so here is the pathway, and this just shows you like a little bit more details of what's going on. So again, we started with our pyruvate. So we had alanine, we have lactate, we generate pyruvate. We're in the fasting stage, so pyruvate will enter the mitochondria matrix. We have plenty of fatty acid oxidation, so pyruvate dehydrogenase will be turned off. Pyruvate will then have to generate oxaloacetate using pyruvate carboxylase, and keep in mind that this required biotin as a carbon transfer. You then generate oxaloacetate. Oxaloacetate cannot cross the membrane, so oxaloacetate can exit through asparagine or through malate shuttles. Now back in the cytoplasm, so out here, remember we're in the cytoplasm, you have either malate or asparagine that are going to regenerate oxaloacetate. Now oxaloacetate can be one of the key reversals is oxaloacetate to PEP carboxy kinase, no, PEP phosphoenolpyruvate through phosphoenolpyruvate carboxy kinase. Sorry, I'm getting all mixed up. This requires GTP. You get rid of one of your carbons because remember oxaloacetate is a 4-carbon, and so you want to generate 3-carbon precursors because glucose is of course 6 carbons. And so now you have your phosphoenolpyruvate. So for pyruvate to phosphoenolpyruvate is you're going to have pyruvate carboxylated, or your 3-carbon is going to become a 4-carbon to oxaloacetate, and this does require biotin. And this is pyruvate carboxylase in the mitochondria that does this. Now in the brain, we do also have pyruvate carboxylase, and this is an anapleretic or refilling reaction of your brain. This is very important, especially for glial cells, and so this is going to help form the excitatory neurotransmitter glutamate. And so the generation of glutamate from oxaloacetate in the brain is going to be key. Now, the carbon or carbon dioxide that was added to pyruvate when it was forming oxaloacetate is going to be released by the reaction of the phosphoenolpyruvate carboxy kinase, PEP-CK. And again, GTP is going to provide the energy, and there is a mitochondria version of PEP-CK and a cytosolic version of PEP-CK, and they're at a ratio of about 50-50. So your oxaloacetate in your mitochondria can become phosphoenolpyruvate, or it can leave the mitochondria in the form of malate. And then in the cytosol, it can be worked on by cytosolic PEP carboxy kinase. Remember, malate through malate dehydrogenase can go back and forth to oxaloacetate. Oxaloacetate through transamination reactions can become aspartate, and aspartate can leave mitochondria, and malate can leave the mitochondria. Oxaloacetate, of course, cannot leave the mitochondria. So keep in mind that our gene transcription is going to be during the fasting stage, and so our glucagon signaling is still going on. And so I just want to remind you that glucagon signaling can increase transcription of genes, and some of those genes in your liver is going to be important for gluconeogenesis. So remember, we have glucagon, and this is going to make cyclic AMP, and this is going to activate pKa. Now pKa is going to trigger Krebs, so it's going to phosphorylate Krebs, and Krebs, if you have trouble remembering what it is, it's cyclic AMP response element binding protein. Now this equals a transcription factor. Okay, so this transcription factor is going to go to the nucleus. And of course, it's going to turn on genes. One of those genes is going to be PEP carboxykinase. Just as a side note, the CREB is going to bind to a Cre element, cyclic AMP response element. And the binding of CREB, we'll make CREB pink, to the Cre element will turn on genes such as your PEP-CK. It'll turn on, of course, other genes that are important for the fasting stage as well. But that's just an example and a reminder of this is being controlled still by hormone signaling. In the case of fasting, that would be glucagon. Okay, so I just talked about oxaloacetate. Oxaloacetate does not cross the mitochondria membrane, and so it has to become something else. It can become malate, for example, and cross the mitochondria membrane. So that just brings back our two shuttles. So we have our malate, oxaloacetate, aspartate shuttle system, and that's just shown here. And keep in mind that this reaction is a transamination reaction. You're just simply moving around nitrogens. And so your glutamate is donated a nitrogen to oxaloacetate to generate aspartate. And it can go in the reverse direction. Aspartate can donate the nitrogen to alpha-ketoglutarate to generate glutamate and oxaloacetate. And then on the other one, you have the dehydrogenase, which is going to be reversible. Oxaloacetate will oxidize your NADH and generate malate, or malate will reduce your NAD and generate oxaloacetate. So keep in mind also that PEP carboxykinase is going to be either in the mitochondria matrix, so oxaloacetate can be decarboxylated to form PEP in the mitochondria matrix, and that can leave, or it will have to go into the cytosol and worked on by cytosolic PEP carboxykinase. So for your gluconeogenesis, your rate-limiting step, your rate-limiting step is going to be catalyzed by your PEP carboxykinase. So going from the 4-carbon to our 3-carbon precursor is the rate-limiting step for gluconeogenesis. All right, so for gluconeogenesis, sorry, I don't know why those images are there. So we have, again, on glycolysis on one side, and we have our gluconeogenesis on the other side. And once you have PEP, you can go both directions until you get up to fructose 1,6 bisphosphate. Same thing with glycerol. Once you get to glycerol 3-phosphate, you just simply reverse everything to go to fructose 1,6 bisphosphate. And then it's at that fructose 1,6 bisphosphate where you have your next major step to reverse glycolysis. So all of the steps at this point are going to occur in the cytosol. So we're going to convert phosphoenolpyruvate to fructose 1,6 bisphosphate. And then these are all going to be reversed. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are going to condense to form fructose 1,6 bisphosphate. And this is the reversal of that aldolase reaction. And so this can be done. There's no huge leaps that have to be made at this point. Now the second bypass is going to be where you have to take fructose 1,6 bisphosphate and generate fructose 6-phosphate. So in this case, this is reversing the rate-limiting step of glycolysis. This is a hydrolysis reaction, and it's going to be highly regulated. So in glycolysis, again, you're going from fructose 6-phosphate with phosphofructokinase 1, PFK1, to fructose 1,6 bisphosphate. And for gluconeogenesis, you want to go in the reverse. And so this requires fructose 1,6 bisphosphatase, and it's going to release one phosphate. So fructose 1,6 bisphosphate releases inorganic phosphate. This is not the reversal of PFK1 reaction. ATP is not going to be produced in this reaction. There's no energy made. The enzyme is fructose 1,6 bisphosphatase. And then we have fructose 6-phosphate, which will be converted to glucose 6-phosphate by the isomerase used in glycolysis. And that's a phosphoglucoisomerase. PFK1, again, is going to be inactive due to low levels of AMP. And it's also going to be inactive due to low levels of fructose 2,6 bisphosphate. And high levels of ATP. ATP are caused by, of course, your beta oxidation of your fatty acids. And so PFK1 is going to be inactive, and so that means that when you reverse, you go in the reverse direction, fructose 1,6 bisphosphate to fructose 6-phosphate, PFK1 won't reconvert fructose 1,6 bisphosphate, for example. Okay, now in your glycolysis, you have glucose 6-phosphatase hydrolyzed phosphate from glucose 6-phosphate. And again, this is not the reverse of the glucokinase reaction, since ATP is not going to be produced either. So remember, for glucose to glucose 6-phosphate, you used up an ATP. Going in the reverse direction, you're releasing an inorganic phosphate. You are not regenerating ATP. And so glucose 6-phosphatase is simply just removing that phosphate. Now, glucokinase levels are going to be inactive because of the lack of insulin signaling as well as low levels of glucose. So it's not going, once you generate glucose, glucokinase will not reverse that and make glucose 6-phosphate again. Okay, so let's talk a little bit about gluconeogenesis precursors. We talked about lactate, alanine, different amino acids, glutamine, and glycerol. Now, lactate is going to be formed all the time through the Cori cycle, and also by muscle cells that are exercising. Through anaerobic glycolysis. Alanine is important because alanine is going to be able to pick up excess nitrogen from tissues, especially skeletal muscle tissues. And so there is something called the glucose-alanine cycle. And so basically the glucose-alanine cycle, just very briefly, is you have muscle cells that will use especially your branched chain amino acid. For energy, to use their carbon for energy sources. And so you have branched chain amino acid transferases in your muscle cell. And remember, this requires vitamin B6 to do these transferases. And basically, you're taking the nitrogen from these branched chain amino acids, and then you're going to pass them off. And in this case, you're going to form glutamate. And the carbons are go to energy for the muscle cell, of course. And then for glutamate, you have alanine aminotransferase. And alanine aminotransferase, again, you need vitamin B6, and this will generate pyruvate. And then this can go to energy. And you also are going to form alanine. And so alanine, of course, picked up those nitrogens from glutamate, and they're going to transfer those nitrogens to the liver. So inside the liver, you're going to have alanine aminotransferase again. And then this is going to convert alanine to pyruvate. See if I can fit this. So you need B6 again, and you're going to get pyruvate. And then this is going to go to gluconeogenesis. Okay, so that's kind of what's happening. And then the nitrogen would go to the urea cycle. And so this is where you can shift around nitrogen so that you get it to the liver cell so that you can dispose of the nitrogen through the urea cycle, and also use the carbons in your proteins during the fasting stage for energy as needed. Now also amino acids can be precursors. All amino acids, except for leucine and lysine, can be degraded to different TCA intermediates. They are then going to be converted to oxaloacetate, then to pyruvate, and then pyruvate can be used in gluconeogenesis pathway. And glutamine is another important amino acid for gluconeogenesis, partially because glutamine will bring its carbons to your kidney. Your kidney does need to do gluconeogenesis for its cortical for the cortex, the cells in the cortex. For the cells in the cortex to do gluconeogenesis for the medullary cells, and your small intestine also does have a small role in gluconeogenesis. So glutamine will go to the kidney and plus the small intestine. So glutamine is the sole source of carbon atoms for gluconeogenesis in the kidney and small intestine. And then glycerol, of course, is going to come from your fatty acids being released from adipose tissue, and that is, of course, going to go to glycerol 3-phosphate through glycerol kinase, and then onwards to form glucose. So in the intestine, you can do some gluconeogenesis. Remember, this is not to maintain blood glucose levels. Glucose during the fed stage will be taken up through sodium-dependent transporters, and then they're going to enter the enterocyte. The enterocyte can upregulate GLUT2 receptors on the luminal side, and that can also take up additional glucose. GLUT2 is always going to be present on the basolateral side of the enterocyte, and the intestinal cells can use glucose as energy and produce glucose via glutamine. So in the intestine enterocyte pathway, you basically have your intestinal cells. So let me just draw a line. So here's your enterocyte. And then you have your blood. And then we'll just put liver here. Or you can have kidney. We can do kidney, just to be a little different. Okay, and so you have in your blood circulating, you have your glutamine. And glutamine can be picked up by your kidney, and it can be picked up by your enterocyte. And it kind of has the same fate in both locations. Of course, the nitrogen from glutamine, should be an N. The nitrogen is going to be used to buffer urine, so that's different than your kidney versus your enterocyte. So in your enterocyte, your glutamine will be picked up and will form glutamate. Glutaminase, so you get glutamate. And then this can form alanine. And then alanine, of course, can be released and go to the liver. Alanine always goes to the liver, so it'll go to the liver. An alternative is the nitrogen can also go to the kidneys. But regardless, you have glutamate. And then glutamate is going to become alpha-ketoglutarate. And this is going to be a transamination reaction with pyruvate. And you're going to form alanine. So remember, glutamine is going to have 2 nitrogens. And so you have to deal with both nitrogens. So this is going to have 1 nitrogen. And then the second one is going to have 1 nitrogen. So now you've gotten rid of both your nitrogens. And they can package them into alanine, and then alanine, in both cases, would go to the liver. Your alpha-ketoglutarate will go into your mitochondria. So here's your mito. And then you can go into your TCA, and this can become oxaloacetate. And then in the mitochondria, you can have PEP carboxykinase. And this can generate PEP. And then this can go on to make glucose. And so in both your intestinal enterocyte and your kidney cells, they both can do this process. Your kidney cells will, your enterocytes, remember, have the GLUT2, and so glucose can leave. And your kidney cells are going to use that glucose for the medullary cells. It will release it into the medulla. Okay, so renal gluconeogenesis is going to pick up glutamine from skeletal muscles. So during the fasting stage, the skeletal muscles will produce a lot of glutamine. And remember, glutamine carries with it 2 nitrogens. And then your renal cells want to pick that up because it's going to help buffer the urine. Glutamine is going to be the preferred substance for gluconeogenesis in the renal cortex. Again, is to get those nitrogens out. It's going to be broken down into alpha-ketoglutarate and ammonia. And then the ammonia is going to spontaneously iodize to ammonium ion, and that's going to buffer the urine. Renal gluconeogenesis can provide glucose during severe hypoglycemia when you get liver failure. However, it cannot maintain glucose levels for very long. Your liver really needs to be, or it really is the major organ to maintain blood glucose levels. Okay, so we talked about gluconeogenesis. It occurs mostly in the liver. There are other areas that can occur, small intestine and the kidney, but they cannot compensate for liver loss of function of the liver. The precursors are going to be lactate, glycerol, and amino acids. Alanine is a very important precursor in the liver. Glutamine is going to be an important precursor in the gut and in the kidney cells. You have 3 major reversible pathways for gluconeogenesis. You have your pyruvate carboxylase and phosphoenolpyruvate carboxykinase to bypass the pyruvate kinase step. And then you have your fructose 1,6 biphosphatase, which bypasses the PFK1 step of glycolysis. And then glucose 6 phosphatase bypasses the glucokinase step.